Shortly after 2 o'clock, so I think we'll get started. Please join me in welcoming Dr. Annie Kathuria. Dr. Kathuria is an assistant professor at Johns Hopkins University, where she joins us from today. Before she'll tell us about all her research, I'm not going to explain what you've been doing, although you've been doing a lot of different things. Besides what she's going to be telling us about today, you've also been working on a number of other organoid types. You were telling me over lunch. Yeah, I'll leave it at that for now. But organoids are your main focus research-wise, but you're also working on translating that into actual entrepreneurial ends. Yeah, translational like to the bedside. Sorry. Anyway, before moving to Johns Hopkins, Dr. Kathuria was at Harvard for a couple of years. She obtained her PhD from King's College in London, and did her undergrad at University of Minnesota. So please join me in welcoming Dr. Annie Kathuria as the 2025 URI keynote.
Thank you all, and thank you for a very warm welcome. So today, as Dr. Eden pointed out, that I'm going to talk about mostly organ-like structures or organoids. And my lab focuses on these organ-like structures, and we've made various kinds. I'll go through one major type, then I will showcase some of the other types that we've generated and made them what is colloquially called assembloids. I don't like that term, but fusions of different kinds of organoids to make something much more advanced, as I would say. So the goal that inspired all this organoid work was to develop new therapeutic targets for neuropsychiatric disorders. And neuropsychiatric disorders are one of the most understudied disorders right now. Well, we're moving towards more mental health, but in understanding mental health, we also don't understand the biology behind mental health. And we are using like organ-like structures to translate this research and improve child as well as young adult health. So 1 in 10 children or young adults experiences severe mental illness, from anxiety to very severe autism to schizophrenia, bipolar disorder, and various others have been studied or found in young adults and children. And it's not like a one gene, one cell disorder. These disorders occur throughout the brain. They have various genetic variations associated with it, but also environmental factors associated, which can happen prenatally, once the baby's born, and then postnatally as well. And these affect the epigenetics and the genetic mechanisms of brain development, and then affects the modifications in the brain, including the amygdala, the cortex, and the hippocampus, and various other behavioral dysfunctions are seen. So we need to develop better models that closely resemble the human brain. And in this case, you can see as an example, and this is the most common animal studied in most biology, is a mouse or rat model. And you can see the difference between a mouse brain versus a human brain. There's a vast difference. Furthermore, you can say a mouse can get depressed because of loneliness, and we can cause it, but I don't think it gets schizophrenia or bipolar disorder. So the drug discovery for this field, particularly for neuropsychiatry, has been severely delayed, but also the clinical attrition rate, which means that before a drug is given to a patient, before it even like goes to phase 1 trial, it's a 96% failure. That high. Neurology is 90%, but 96% for psychiatry is very, very obvious now. You can see why, because we need better models that can at least try to mimic some part of the human brain. And this leads us to the concept of induced pluripotent stem cells. So induced pluripotent stem cells. So show of hands, how many people know what induced pluripotent stem cells? Great, just a recap. Yamanaka in 2012 won the Nobel Prize for it, and in 2007 and 2008 they made this. They started with the mouse, so they took some skin cells, fibroblasts, and revert the age of it by using 4 transcription factors. They're now called the Yamanaka factors, and with that they converted its age back to a stem cell. Next year in 2008, if they said they could do it with mouse, they did it with humans. So 2008, they published it in the human. So now you can take hair cells, skin cell, blood cells, even urine cells from urine. Anything you can think of most of the time, they can use that to revert back the age to a stem cell. What it gives us the advantage of doing is with this stem cell, it is personalized to you. So if I take my hair cell and convert it into stem cell, it is personalized to me, it carries my DNA signature. So imagine doing disease biology and disease development with a schizophrenic patient who has been diagnosed with schizophrenia. We don't know which gene is affected, but we can take that patient's hair cell or blood cell and convert it back into stem cell. Now it is personalized to them because it carries that genetic makeup. So with that in mind, people have now started developing this whole organoid field and 3D tissue engineering field has been. My focus has been brain, but we've seen retinal organoids, skin organoid, kidney, stomach, intestine. Yesterday at my class, I was teaching lung organoids. I was like, wow, that also exists. Pancreas. I was in the Biomedical Engineering Society conference in October. I saw salivary glands, and I was like, wow, okay. So there's all kinds of organoids, and this field has expanded massively. So what are we using these organoids for? First of all, we can learn a lot about developmental biology. So how our human body develops. We get to do a lot of disease modeling, as I mentioned, with personalized, so we can do disease modeling, control versus a schizophrenic patient, control versus an autism patient. And then drug evaluation, which is what primarily my lab focuses on with neuropsychiatric disorders, drug evaluation. We can basically do like a clinical trial in a dish on these organ-like structures and find out what drug we can use. That's the translational piece. And people, which is the moonshot these days, I say, are also focusing on is transplantation in cell therapy. So imagine transplanting these back into humans. And that has also been going on in the next 5 to 10 years, you will see that. So for the purpose of this talk, I will focus on 4 aspects. Bioengineering the cerebral organoids, so the cortex region that makes you who you are, gives you the ability to think, talk, make decisions, sometimes good, sometimes bad. Cerebral organoids, so how do we characterize these? How do we know that the cell that we have generated is the cognitive or the cerebral cortex that we generated? And then disease modeling, so example of disease modeling, and I'm known for schizophrenia, and we have schizophrenia patient-derived cell lines. So I will talk about how we use cerebral organoids for schizophrenia. And then the last bit is what's next, or what kind of organoid how can we advance the organoid field, particularly in the brain region. So for bioengineering the cerebral organoids, we start with the iPSCs, so the induced pluripotent stem cell. They were very 2D in this time point. We grow them into this 2D adherent layer. What we do with it is the first week, we try to push it towards the ectodermal lineage, which is the lineage that gives neural tube formation. So the early neural tube formation starts. And then with the neural tube formation, we kind of aggregate these into ball-like structures, which we often call in the lab embryonic bodies. And once these embryonic bodies are generated, we encase them into these extracellular matrices, which provide support and network systems to these cells to grow and expand. And lastly, it takes, I'll show you the whole detail of it, how these organoids are formed, and then we make these cerebral organoids. So the fun aspect in this is the fun picture I like to show often enough. This is a picture I took with my older iPhone, but it is in the lab. It's a 6-well dish with little pea-like structures. So this is an organoid. I would say these are small little pea-like structures. The running joke in the lab, and one of my students asked this, I don't know if you will laugh at it or not. Can we eat these? Can you make it smarter? And I was like, we're not eating them. It will not make you smarter, not intensely. And why did we want to eat them? The other question was also, this was another fun one. If I eat this, is this cell cannibalization? And I was like, please, let's not go there. Because it's human tissue, and I was like, okay. The questions I get. So going back to the science, be serious. Take the iPSCs. So we take the iPSCs, and this is right now in 2D, as I mentioned. We force it to the ectodermal lineage, and these are some of the factors that we use to add, and it takes us a month to generate these. Actually, now we've gotten even shorter, but when I first started, it was about a month, and we add these different factors to cause the lineage. So SB43 is an example of a TGF beta inhibitor, so signal, remember your biochemistry pathways. Noggin is another one which inhibits BMP signaling, so another factor adding. Then I add again to SB43, which inhibits at this time point active in the nodal pathways. And then again, Noggin was added again to inhibit BMP. The whole thing is to get this ectodermal lineage, with the idea that once you get the ectodermal lineage, you start seeing some of these positive markers for ectoderm, which I will show here. So right now, everything is in 2D. How do we make it into 3D? Is we dissociate these cells. We dissociate them and then aggregate them using just a centrifuge. We use a dissociation agent. We call it commonly acutase, which is a very gentle EDTA. And it splits the cells into single cells. We aggregate it using the centrifuge, spin it around, and these little ball-like structures are called ABs. And they look something like this. So we get hundreds of these, and you can see their SOX2 positive, which is a neuroelectrode marker. So once we start with these, we started like hundreds of these, and then as they grow, they could become a little smaller, or they kind of fuse together. And the most important step is encasing these emerald bodies into the extracellular matrix. Now, why is the extracellular matrix important? The extracellular matrix is important because it contains these membrane proteins. These are the proteins that help think of a skeletal system for your cells, provides the glue and the skeletal system for the cells to expand and grow and form networks. And in this case, we use a combination of laminin, fibronectin, and collagens to encase these. Depending on the tissue you have, this changes. So if I'm trying to make the cortex, this is the tissue I would use, or the extracellular matrix I would use. But if I'm trying to make heart organoids or blood vessel organoids, this will change completely. And so it depends on what tissue you have and why you need that tissue. And that depends on your disease model as well. So this is what a 6-month-old organoid looks like on the right-hand side corner. And the scale bar there is 1 millimeter. So this is 3 to 4 millimeter in size. As I said, they look like little pea-like structures. And we often see them not grow after 6 months. So after 6 months, they stabilize, and this is the size they will remain at. So now you know how we made the organoids. And now the next part is, once you make tissue in the lab and you're actually engineering these organoids, what does it look like? What is it we've generated? Did we generate the cortex, or did we generate something else? And in this case, we characterize them using immunohistochemistry, as well as some genomic analysis like bulk RNA sequencing and single cell RNA sequencing to see what cell types are we getting. So first we started with characterizing them just by phase contrast imaging, just the size. And again, this is another 6-month-old organoid around 4 to 5 millimeter in diameter. So we did this at 1 month, 3 months, and 6 months. So what I would like you to see in this graph is the bottom x-axis here has these number ML15. These are just cell line numbers. What I want to showcase here is it's agnostic to disease lines. So it's a mix of control, bipolar, schizophrenia patient lines. It doesn't matter which patient-derived line it is from. The cell lines remain, or the organoids produce the same kind of organ-like structures every time we reproduce them. So reproducibility in these organoids has been better with our methodology. This is the current challenge with the organoids right now is reproducibility, and we have shown with agnostic of the cell line that we have, they do reproduce at the same kind of size and shape-ish, roundish structure. Not completely round, it has some roundness is not the best issue. I would say the best would be the size. And so they start at around 800 microns too, and then at 6 months, they're around 4 or 5 millimeters in size. And they don't grow after this, after they stop growing. So now what is inside the cells is important as well. So here, this is another 6-month-old organoids. Much of them are 6-month-old organoids. Again, these all colors represent different cell lines. So they are different cell lines here. And we are looking at different markers. Each marker has a significance. So example, CTIP2 is a layer 6 cortex marker. GFAP, which is an astrocyte marker, SATP2 and PUX1 are layer 2-3 cortical markers. And the fun ones that we found were this myelin basic protein and oligospecific protein. This means we are producing oligodendrocytes, which means they're producing myelin. And if myelin is being produced, basic neuronal biology, electrical conductance is better. That's what's the function of myelin. And with that, the other thing we saw was neoprogenitor cells, which are PAC6 cells, so neural stem cells that form more neurons, and TBR2, another neuronal progenitor marker, which is also being produced, which turns into TBR1. TBR2 turns into TBR1, which is another layer 6 marker of the cortex. We get inhibitory cells in here as well. LH6 shows that, and glutamate synthesis is a generic excitatory marker. So there's a lot of excitatory neurons, which we would expect in a cortex to produce. We also see presynaptic markers, which I haven't shown here, but they are on the immunohistochemistry with the cross section. So bassoon is a presynaptic marker, as well as Homer here is a postsynaptic. So we get synaptic markers as well at this. So if we get synaptic marker, think about it, it would produce electric current, and a brain is not considered a brain until it communicates via electrophysiological current. So keep that in mind, I'll come back later, I'll quiz you. So we confirmed all over again with single cell RNA sequencing. And this is at an earlier time point at 3 months of differentiation. So we see the same kind of rate. So the ratio is changing this time because it's younger and earlier in development. So it has glial cells, neoprogenitor cells, inhibitory cells, and excitatory cells. At this stage, it has more of the excitatory and inhibitory neurons and progenitors. As the organoid develops, it has more of the glial cells appears at later stages. And this is an example. I'm hoping the video works. Try without the pen. Oh, yep, and it did work. This is a light sheet microscopy. So we deliquefy the organoid and try to do light sheet microscopy. The idea was to just check the general structure of the neuronal. So the neurons are on the outside and the neoprogenitors on the inside. That has been the usual structure that we see in these organ-like structures. And we haven't seen, we see sometimes cortical layering, sometimes not so much. we're trying the different protocols to get very consistent cortical layering. But what we see is a very definitive cell types, which is very consistent between organoid to organoid and line to line. But here, that is that the neurons are very dense on the outside, and the neuroprogenitor and neurostem cells are on the inside. So how closely can we mimic the human prefrontal cortex? Because our target was cerebral cortex, we wanted to see how close we can get to the cerebral cortex. And is there an advantage of having 3D versus 2D? And so we took about 8 control healthy lines and differentiated them in 2D, just 2D excitatory cortical neurons, and then 3D organoids, and started comparing those two with each other, and also compared to post-mortem human data that we have seen. We got this data from the Brain Allen Institute. They have a lot of gene expression data available. When we did it, it was not available. Now they have made it public. So we cross-compared it to various stages of the prefrontal cortex development, particularly only genetically, not in any other way, just the gene expression. So how many genes are being expressed? How many of them are common? So we did it at 8 to 9 weeks post-reconception. That's PWC means post-reconception there, 13 to 15 weeks post-reconception. So all in utero brain samples, and 19 to 24 post-reconception, and then an actual baby that is around 0 to 5 months old. And we cross-compared our 2D neurons that we make from iPSCs and prefrontal cortex or cerebral organoids, those, and then the prefrontal cortex with the whole prefrontal cortex cross comparison. We saw that the most match it happens to is the organoid at 0 to 5 months of development. So you can see the organoid matches significantly, correlates to it, to the whole prefrontal cortex at 0 to 5 months. And this is a 6-month-old organoid too. So the age is matching, at least it correlates well. And then the 2D neurons didn't correlate as much as they should have. We also checked for, which I haven't shown here, is neurological processes like synaptic development, synaptic plasticity. Those are the things we concern about when we're looking at autism or schizophrenia, those kinds of disorders. That had a higher correlation of significance of 2 stars instead of 1 star. So that was more correlative. And in that case, the neurons, the 2D neurons, weren't that correlated. So more correlation was seen with the organoid versus the 2D, and the age was roughly around 0 to 5. The best correlation was seen. So now I have talked about the organoid. We produced the cerebral organoid. We characterized these cerebral organoids. And next is disease modeling using the cerebral organoid. How do we use it in actual test cases, cerebral organoid? And so schizophrenia, you might have known, or you have learned about it, you might have seen TV shows, and so it causes delusions and hallucinations and disorganized speech and paranoia. All those are the positive symptoms of schizophrenia. The negative symptoms that nobody talks about is lack of initiative, the ability to take decisions, lack in speech, dysregulation in thought processes. So it has a cognitive side to it. And that cognitive side does not have any drugs. There is no way the negative symptoms, or sorry, the positive symptoms of delusion, hallucinations, there's drugs for that. Those are also sort of effective, but for negatives, there's nothing for the cognitive side of function of the brain. So with that in mind, we started looking at the cerebral cortex development of these organoids. So we made organoids from 8 healthy controls and 8 schizophrenia patients. These are idiopathic in nature, which means that they don't have a particular gene mutation that has been associated with schizophrenia. They just were diagnosed with schizophrenia. We took their fibroblasts, we made iPS lines from them, and we had 8 patients. So there was an 8 by 8 comparison The second step is to transcriptomically compare the RNA sequencing and look at pathway analysis generated from these transcriptomics and look at the differentially expressed genes, which we call VGs, with known databases, known pathway analysis, and derive some kind of hypothesis, like what pathways are going wrong in these patient lines. And with that in mind, then look at functional cellular molecular phenotypes that could be associated with schizophrenia-derived patient organoids. And then potentially the next step of this would be, once you have a cellular molecular phenotype, then you can use drugs, known, unknown, whatever, to ameliorate those effects. So we did a whole transcriptomic analysis. It's a very intimidating graph and very colorful and makes no sense whatsoever. And I don't even know what to make sense of. What I try to showcase here is, don't get intimidated with color. What the significance of this is, there's upregulation in the purple and downregulation in the yellow. What you want to see is, oh, are the schizophrenia behaving like the schizophrenia, or they're segregating, they're not overlapping with the control, and the controls are behaving and aggregating or clustering together, which is what we see here. The controls are clustering towards each other, the schizophrenias are aggregating with each other, and there's a huge difference between the two. That was the most important part of just these heat maps, is to see if we can segregate between the two patient populations, and we can. The next one was GWAS. So GWAS is genome-wide association studies. So people have looked at schizophrenic patients, their samples, blood as well as postmortem brain samples, and looked at what gene loci in the genome, how many loci are affected. So they have reported about 108 genome loci that are associated with schizophrenia, or those genes associated in those loci are affected in schizophrenic patients. So there's a whole list of these. And once they have the list, what we did was look at a control versus schizophrenia, differentially expressed genes. We have thousands of these. There was 1,000, around 2,000 something, I forgot the exact number, but it was over 2,500-ish. And we wanted to map it with the GWAS associated genes of schizophrenia known. The point of the goal was that, is there overlap that the schizophrenia associated genes that we see in the organoids are also appearing in the schizophrenia GWAS genes? And there was, this is a very short list because I don't have enough space here, but it was 56% of our differentially expressed genes matched with that. And these are some of the top 20 or something we show that were dysregulated. With these, what we started to do, these differentially expressed genes, we started to look at, pathway analysis. There's a lot of pathway analysis and databases that exist. There's Hallmark, Reactome, and there's always differentially expressed gene markers. So we started looking at this pathway analysis, and we did something called gene set enrichment assays. This was done with the help of the Broad Institute when I was there. They have these gene sets in different pathways, and then we started analyzing those. The point is what of these pathways are, in comparison to the control healthy organoids, are dysregulated. And there's some that I'm going to highlight in red, which are the ones that you want to look at. Is, as I mentioned previously, there was myelin in these. So that means, and I showed synaptic plasticity markers or synaptic markers like bassoon and Homer. So there is issues with, here you can see some of the chemical synapses, reactant transmission, chemical synapses were dysregulated, long-term potentiation was dysregulated. So there is some synaptic issues with the schizophrenia. So keep that. The other one that was important and I like to highlight is the oxidative phosphorylation, which was a new pathway that we did not expect to see. We were expecting synaptic plasticity in schizophrenia being dysregulated. GABA synthesis, which is the inhibitory pathways, has been associated with schizophrenia very previously in postmortem studies to be dysregulated, and it works. So that not only verifies and confirms, but something new that came up was oxidative phosphorylation. That was a dysregulated in schizophrenia. So oxidative phosphorylation is very much associated with oxygen consumption of the organoid, and brain consumes a lot of oxygen because it's the most functioning organ of the body. You have to think all the time, right? So with that in mind, we wanted to look at mitochondrial pathways. Mitochondrial pathways that could be associated with schizophrenia. So there is a database in the Broad Institute is called mitogenes. They have mapped all the human genes associated with mitochondria and what are the functional pathways associated. So we took all those genes, and then we took all our differentially expressed genes between the control and the schizophrenia, and we started mapping those together to find out pathways in the mitochondria that were dysregulated. And there's these three pathways that you can see. Downregulated in schizophrenia means they were downregulated, so they were lower, or their genes where expression was lower, upregulated means high expression. And that's in schizophrenia. So we saw all these three pathways, and they were all associated with mitochondrial function and mitochondrial pathways and oxygen consumption pathways. So this gave us the idea, okay, we need to test for oxygen consumption. And we use this assay called seahorse assay. Seahorse is by Agilent technology. What it does in a nutshell is you have different stressors for the mitochondria, all these chemical stressors, and then you measure the oxygen consumption rate of the organ that you're testing. And in this case, we're testing cerebral organoids from healthy patients as well as schizophrenia patient-derived organoids. And you can see we measured this, and there's this significant difference between the control and the schizophrenia. So this was a new sort of finding that mitochondrial dysfunction is very much been associated with neuropsychiatric disorders, but this was particularly for schizophrenia. And now there's a whole mitochondria field that has developed in the last 5-6 years on mitochondria and mitochondrial dysfunction associated with not only the schizophrenia, but bipolar disorders and others as well, even Alzheimer's disease. So what next was, I told you about myelin and electrophysiology, now it's the quiz time. So we actually looked at electrical conductance. We placed these organoids onto multi-electrode air chips. And these are well plates that have 24 or 48 well plates. And then we put these little organoids, stick them on, and record the electrical current. Hopefully the video again will work. This is an example of a control video of just electrical activity, spike activity being recorded from the organoids at different stage. But this is an example of spike activities in raster plots. Shows that we can record electrical activity of these organoids. We do them all the time. We have a very standard method of doing them. The more the mature the organoid is, the better electrical activity this is. So the longer we grow it, the more activity we see. And so what does it mean biologically, all these activities? So I'll give you a very short description of it. So every time there's a spike that is showing in this raw voltage data, that single spike is considered as the frequency. We don't consider that's the frequency, mean firing rate, that's the frequency. That's how active your organ is, how on baseline conditions is producing these activity, just normal action potentials. But then what we wanted to look at is this advanced thing is called all these markers that the MEA field or electrophysiology field has generated. So interspike interval. So if it's spiking so fast, the frequency is higher, mean firing rate is higher, interspike interval is lower. So they have an inverse relationship. Then burst happens. So if a repetition of spikes repeats itself, it's called a burst. And then it has characteristics of its own. How many spikes in the burst? What is the duration of the burst? And interburst interval, how long does it take to burst from one point to another? And then you guys start getting these synchronized patterns that appear, which become the wave patterns of the brain that have been associated, say, delta waves, gamma waves. But we're not that far yet. We just get some spike activity and some interspike interval and some burst frequency that we can record and measure between a cross comparison between a control and schizophrenic patient-derived organoid. We've done this in a lot of organoids. So this is an example. I showed you we could measure bassoon and Homer. So this is an example of measuring that and seeing that there was baseline differences. And with that, we characterized all these different parameters. We not only did this for schizophrenia patient-derived, but we also did it for bipolar patient-derived organoids as well. So cross-compared all these, and every patient has some sort of difference. Bipolars react a little differently to the activity, or showcase different activity, while the schizophrenia have a different electrophysiological parameter. Now, how do we translate this into a translational model? With the oxidative phosphorylation in mind, there has been schizophrenia patients, a subset of schizophrenic patients that show in the blood, they have taken blood draws from them, and in postmortem studies show oxidative stress as a marker. And it's glutathione that they've seen, which is lower in their blood. So a subset of schizophrenic patients we had only an 8 population, and we started going back to their blood work. How many out of these 8 patients had that oxidative stress marker that people have been looking at? And we found 5 of those that had lower levels of total antioxidants and glutathione in their bloods. So we looked at that and we said, okay, so those could be the patients we can look at, because oxidative stress has also been associated with targeting and is sensitive to inhibitory neurons. Particularly inhibitory neurons that are very sensitive to oxidative stress have been seen in postmortem brain samples of schizophrenia too. So we saw that, and with that, we wanted to find out if there's some kind of drugs that can protect the inhibitory neuron. And so while we went from 3D cultures to very 2D cultures, we went from a very complex model, which I think is complex, but can be a definition, and to a very simplistic 2D neuronal marker assay. And we started measuring the inhibitory neurons, just high contenting and measuring the oxidative stress. And you can see that the schizophrenia organoids, or not some organoids, inhibitory neurons show innate oxidative stress. And this is just a very simple assay that measures fluorescence markers for raw species, so reactive oxygen species. And so we saw a decrease in that. And then we started giving them all kinds of drugs on it. The one that I like to talk about a lot, I can't mention you, I was working with a company, what drug they gave, I have no idea myself. We tested their drugs, but one of the drugs that I would put on, which is not actually a drug, you can actually go to Whole Foods and buy it, or Amazon. It's called an acetylcysteine. It's in vitamin. You can actually, and it's found in blueberries, so eat a lot of blueberries. It is an antioxidant. It was used during COVID a lot. It was on my mind, we were doing these experiments at that point. So we gave a lot of N-acetylcysteine to these inhibitory neurons, and they did show neuroprotective effects. And actually, this was then, I was told that I should tell this to the psychiatrist. So we gave all this data to the psychiatrist who really was seeing these patients. And I think one of the patients took it and has been doing better. So we went from somewhere between an organoid to somewhere giving a drug to a patient. Well, not a drug, but some kind of vitamin that helps or has a helpful effect or a neuroprotective effect on the patient's life. So, we saw bioengineering cerebral organoids. We characterized this. I showcased you the cerebral organoids with schizophrenia. The last one is, can we make the iPhone 15 version of the organoid? So this is what we did recently. This is in my work at Hopkins. So once I got into, I would say I'm a very big addict of tissue engineering now. So I like to make organ-like structures. It's kind of fun for me. And now play a little bit of Legoland, playing things, putting things together. So we started with the cortex. You saw the cortex. We then next generated brainstem, which is the mid-hindbrain. And I wanted to stitch it all together with something that could interconnect the two organoids, not just fuse them together because we want to fuse them like an assembloid. I want them to be interconnected with some kind of system. And for that, we generated endothelial organoids or blood vessel organoids. So, and that generated what we called the multi-region brain. And we started from the same iPSCs. So everything was started from the same healthy control line. We made the cerebral, that's shown in yellow. The endothelial organoid is reddish, and the bluish is the midbrain, hindbrain, which we now call brainstem organoids. So what is so special about this? One thing that you might ask this question as well. As the cerebral organoids grow 6 months, 9 months, 8 months, whatever long, they start dying from the inside. There's no vascularization. And that was the idea of generating these endothelial organoids. Not just connecting the two regions, but also vascularizing from the inside. And we said most people or most publications you will see use human umbilical endothelial cells and just add those. They forget that the endothelial cells or blood microendothelial cells do form little tubes, but they need support systems for those tubes. They need the smooth muscle cells. They need the macrophages to form the immune system. And they need these parasites to form the blood-brain barrier. So all this is required, and it's not just one cell that makes the tubes. It is the whole system that makes the tubes. And that's why we generated these endothelial. I can't take credit for this. There's a paper that started making blood vessel organoids. We modified that paper to make ours compatible with the brain because not all cells are compatible with the brain. So we had to make the ones that were more compatible to the brain. They didn't have parasites. We needed to make parasites for the brain to form the blood-brain barrier, or at least start the formation of blood-brain barrier. So we made this, and this we characterized using single cell RNA sequencing, or not single nuclear RNA sequencing. And we see, as I told you, parasites, macrophages, endothelial cells, mature endothelial cells, all kinds of different cell types, all coming together. My favorite picture is on the left side where we injected a dye to see, and we did it took us a while. Like, I think I took me 3 days to take that image, and 2-photon imaging and then 3-photon imaging to get the dye and see the little tube formation. And this is at 2 months of development, so it's not that long compared to the 6 months that I've been showing you. And these are much more advanced because of the advanced structures involved in them. So the multi-region brain organoid with the stitching looks like this in terms of light sheet microscopy. So these blue cells are the endothelial cells or the vascular organoid, and that kind of engulfs the little brain organoid. Both the brain organoid fuse together, which is shown in green, and the pink is the hindbrain marker. And so it's a huge organoid. This is within 2 months, we get about 3 millimeters in size. We are still growing them. They are now 4 to 5 millimeters. So now we've upgraded ourselves from small peas to small blueberries. That has happened, but we have not been able right now to see if we can sustain the dynamic nature of a blood vessel, which means they're not hemodynamic yet. They're not having flow or perfusion in it because I don't have a pump to flow it yet. We're trying to establish that in the lab now. So we did a lot of single nuclear RNA sequencing with all kinds of permutations and combinations. And we wanted to showcase with this multi-region brain organoid, it's not that we're not getting every cell type that we said it's there. So we get midbrain, we get cortical, we get hindbrain, we get a lot of extracellular matrix formation, and endothelial cells, a lot of endothelial cells. With this, the fun part was we saw a lot of early blood-brain barrier formation, which is tight junctions. We see a lot of tight junction formation within the organoid. The sad part is I don't know how to measure the barrier function right now in 3D because it's a very dense tissue. I can measure the dye and this tube formation, but since I don't have flow, I don't know how to measure some kind of way to see if the blood-brain barrier is working or not. So that is pending. We're still working on it. The other way to measure blood-brain barrier is through tear function, and that's trans-electrical resistance. So any barrier you have, you can think of it as a rubber band. You poke it with an electric current, it resists back, and you get that measurement. But that's also done in 2D. We can't seem to be able to do it in 3D, so we're working on it. So we did form, at least with this blood-brain barrier formation of the early stages, we can look at the protein, we look at immunohistochemistry, and we have done a lot of genetic analysis and seen all the markers that are present. So they are present. And if you take the vascular structure out, which is the endothelial organoid, it disappears. So we've done that cross comparison as well. So I like to end with this amazing slide that I love to showcase is no one compares the organoids to an actual postmortem brain or whatever organ they look like or compare to. Very few people do it. And I always like to compare where in the development stage we are at. So the black in there is single nuclear or single cell RNA sequencing of a fetal brain at 40 days. And we overlap our multi-region brain organoid on top of it, the same single nuclear RNA sequencing. We get an 80% cell cluster match, which does not mean it's an 80% match to it. It means all the cell types that are present in a fetus at 40 days of development, we get all that cell types in the same ratio. So we are the closest that someone has gone to a whole brain, at least in the fetal brain. So we get all the astrocytes, progenitors, myelination, oligodendrocytes, all the different kinds of cell types that you see here at an 80% ratio match to an actual human fetal brain. So that was my discovery in last 2 years. And then the hope is to now use this model for neuropsychiatric disorders, for neurodevelopmental biology, and other kinds of drug discovery paradigms. So with that, I'd like to stop talking, and I'll give you the opportunity to ask me all kinds of questions. This is my little lab. I have to change my picture. Most of these students have disappeared and gone to the PhDs now.
I'll start real quick. For your multi-region brain organoid, you said you begin developing each on their own and then bring them together at day 20. And what does that fusion look like?
So we start with the cortex, and then we've developed both the regions together. So we first fuse the cortex with the blood vessels, and then the blood vessels, we have a color in there, we have cherry in there, so fluorescent markers, so we know which area is blood, and then we fuse the other one on the bottom, basically. And that integrates, and the blood vessels, neurons are postmitotic, they don't grow. The blood vessels are not postmitotic, they really like to grow, so they kind of take over and absorb the two organoids within them. Or they will form like interconnections between them.
Right.
Yes.
Building off that, when you have the brain all together like this, these parts stitched together, is there a way to then encourage them to grow after they've been assembled? Or is it kind of like,
No, they grow.
oh, they
They
will
grow.
grow,
They grow indefinitely.
Right.
So this is roughly 3 millimeters in size. They are the ones now, and this was at 2 months of development. So 20 days and then 40 more days of growth. But now we have them 4 months of development, and they're pretty big. They're 5 to 6 millimeters.
And the activity increases as.
Oh, yes, they are very, very electrically active, and the 2 months they are very active as well. I can show you, it's in one of my slides here, that it does show a lot of electrical activity to cross compare with the other organoids. Midbrain and hindbrain stem cells are the most active, no matter what I do. They're very active within 30 days, but the MRBO catches up very fast around 85 days of running. Yes.
You talk a lot about a lot of the activity. Have you run a patch test on any of
Oh, it's
those?
very hard to run on patch. I can't find a little neuron, hoping to. I admire the people who can do it, not me, but we have done it once and it does show, like you said, very nice action potential, but you need it.
And do you work with the Schneider's department over at med school? No.
I am at med school. My lab is in the med school. No wet lab space in the Homewood campus. So we work sort of,
If I wanted to get involved with the organoids, would I apply to Schneider, or would I apply to the friends, would I apply to the regular school?
regular school.
Or do it
We
school?
just call it texting. There is one more organoid lab at Hopkins that's in the public school. Thomas Hopkins. They don't do this. They are cortex. Right.
You mentioned that you couldn't keep, those blood vessels, right? Like, you couldn't keep, stable, right, to continue the. So I read about, 3D printed,
Yes.
bioprinted, like scaffolds. Have you thought about incorporating?
Yeah, well, I just hired a postdoc. She's a 3D bioprinting expert. So we're gonna use the biological material itself to print it and, well, we already have the blood vessels. We don't have perfusion in it. So we're trying to make it so enough strong to get perfusion and flow. So she's gonna do that. Yeah, that's the iPhone 17. Go ahead.
I think this is a simple question, and excuse me if I missed it if you said it, but how long do they live? When do they start dying, or is it kind of?
A cerebral organoid of cortex that the schizophrenia data you show will start dying around 8 to 9 months in development. I have grown them up to a year. They won't die that easily, but around a year, their quality starts decreasing. They get a necrotic core. They see a very dark black spot in them, and that means they're dying. So we usually throw those out. But these have been growing because of the endothelial structure. The endothelial structures themselves, or the vessels themselves, have some sort of innate perfusion of some kind, which I'm trying to figure out what they're doing, is they have been more oxygenated and more healthy.
So you think it's just the lack of oxygen or?
Yeah, lack of
vasculature
oxygen,
that's
yeah.
killing them,
Go ahead.
Just as a follow-up to that, so far the endothelial ones, because they've just been created, none of them have died yet. They're
No.
still growing.
They're still growing. It's about 6 months in the lab already. Yes. Yeah, you had a hand up.
The goal is to eventually like introduce these organoids into patients.
So the goal, there's two folds of goals that we have in mind. The first goal is to do for drug discovery. As I mentioned, we did drug discovery for schizophrenia. You could use the same platform to do drug discovery for schizophrenia all over again and find much better compatible drugs. So that's personalized medicine and the concept of personalized medicine or precision medicine these days, whichever word you want to take. That's one. And the second one is, which is a moonshot right now, is transplants. To put these back into people, and you and I were discussing at lunch, how people can use them for transplant when there is traumatic brain injury or some kind of injury, then remove the scar tissue and put these back. And because the neuron does, once it's dead, it doesn't grow back. So these can be a regenerative therapy.
So my question is, if you're trying to possibly readjust. You mentioned that the ones you currently have are still growing, to me that just sounds like unregulated growth. How can you guarantee that these organoids are gonna respond to other cells around them in the same way as normal cells? If not, you have unregulated growth and possibly.
Yes, that is one of the biggest problems with iPSCs and stem cells is unregulated growth because of tumorigenicity, and that's why the field is very wary of putting them back. Having said that, so the test case that we haven't done this yet, we're in process of doing this. I'm working with the neurosurgery team at Hopkins. They have a traumatic brain injury model for rat and pigs. And the idea there is they're going to do it at a very specific location on the brain. They're particularly interested in cognitive function. And so they're going to injure a very particular area. And then that area, once it's injured and then it develops scar tissue, they will remove the scar tissue. But when they remove the scar tissue, they will know what area it is. So we won't put the whole multi-region brain organoid. When it doesn't require brainstem, I'm not going to put brainstem. Because it's modular in nature, we can use the cortex itself if they're looking at cortex, and we just put cortex in it. And we will put vascularization because this helps with graftability. These little tubes are very useful because they are graftable. Yes, go ahead.
Isn't iPSC still done with virus vectors? So you.
No, not anymore. No, they are just gene modifications now. Think of it like the COVID vaccine mRNA. They've been used to program those now. Oh, initially when I was doing my PhD, it was viruses.
But they're still not really compatible with CRISPR.
No, they are not. They're often. Yes.
Hello, I'm Tom Nunzio, I direct the Rome University Schizophrenia Spectrum Lab, even later today. From a clinical kind of side of things, do you have any information about donors in terms of their symptom profile?
Yes.
And other kind of backgrounds that might,
So-
how does that fit into the specificity of how your mind's kind of reacting?
So, most people will design the study according to what they want. So in this case, I didn't get to choose my lines, but let's say for schizophrenia, they would take patients. So we get de-identified lines. I get de-identified lines. I don't know who they are, what they are, but they will tell you the age, sex, medical history, which is what medications they were on, what medications work, what did not work. So I don't show here, but we did a study on clozapine resistant and clozapine, or the clozapine worked for those patients. And the ones that showed clozapine worked for this, the neurons responded to clozapines. The ones that did not were resistant to clozapine did not respond to clozapine. So that we've done.
And you get their symptoms, so you mentioned the positive and negative symptoms, do you have a sense of how severe?
Yes, they,
Duration of illness? and things
yes,
like that?
they tell us what it is. So the recent study that they're recruiting patients, I'm not, but for like treatment resistant, complete treatment resistance, and had 2 psychotic episodes and then turned treatment resistant. So those patients that have no avenue whatsoever, those they're trying to make iPSCs out.
Any differences? You mentioned the lack of kind of effective treatments for those with negative symptoms. And did you see, so with the clozapine resistant, were those the ones with higher negative symptoms or?
That I don't remember off the top of my head. I had to look it up with.
I just wonder if there's any kind of signal there in terms of some.
Yes, I would look at that. I remember there was this clozapine resistant and non-resistant. And there was no secondary issue, like with bipolar patients, we get a lot of, metabolic issues. With these, there was no metabolic issue. I don't remember that much, but I would have to look at my table.
Thank you.
Yes.
The hand down there.
Oh, sorry.
No, don't worry. So I'm interested in like the risks of, people's risks of developing schizophrenia. And risk factors that, can tie into that. How do you think these like organoids could help, better their understanding of, those who have symptoms and don't have symptoms, and like the risk factors?
It's a good question. So the way the iPSCs, you can say, are very useful is they're a blank slate. So that means I can test for risk factors in more susceptible people. So one of the studies is not schizophrenic related that we're thinking of is a substance use disorder, right? Some people are more prone to substance use than others. So you can cross compare those that are more prone to substance use disorders and some is not, and then test each kind of drug and see the susceptibility of which drug is more. So for example, we're doing cannabis right now. And cannabis does induce sometimes psychotic symptoms in early adults. So, thank you. Yes.
So you mentioned switching between 2D and 3D culture multiple times, and I'm
Yeah.
curious, like, from a purely ignorance perspective, how you do that? And then secondarily, like, what is different about the cellular connections, the connectivity of the neural cells in the 2D cultures compared to the 3D?
So we just have the same lines, we just differentiate them separately, just the technical bits, the first question that I'm trying to answer here. So organoids in general, it's a different process of development and different process and protocol. So we'll just use the same cell line, make the organoid, same cell line, differentiate in a different way, in a 2D inherent way to make the neurons.
So it's pure, there's nothing about a scaffold or the
No.
environment you're giving them, there's no mechanical contribution to whether they're.
Mechanical, no. Environmental, yes, with the chemicals.
Yeah.
So chemical differentiation. Scaffolds on the organoid, no scaffold in the neurons. And then cell to cell communication. I'll give you the best example. Electrophysiology is the best way to measure neural function as well as neural maturity. And so a neuron in 2D or neural cultures in 2D will take a while to show electrical activity. They won't even show electrical activity even if you don't add astrocyte conditioned media or something to help it grow. An organoid will show it. You don't need to do something special to it. It will start developing that. I think it's because of the support system it has with the extraglial cells, the neurostem cells, the combination of having excitatory, inhibitory neurons together. So all of that helps the organoid mature faster and produce more electrical activity. So that's how I would answer that question.
Thanks.
Go ahead.
Kind of as a follow-up to that, I know that the method you talked about for doing this involved growing the parts of the brain separately and then using the endothelial mechanism to tie it together.
Yeah.
I don't know if this is something that you have tested, but would it make a difference to try to grow the three parts together and at the same time as the endothelial?
There are different factors involved in each different.
Part of the
media.
chemical media needed for it.
Yeah.
Okay.
Someone has tried to do that, but they didn't end up getting the whole brain. They just got the prefrontal cortex, but a very advanced prefrontal cortex. Yeah.
Has anyone tried this, the campus?
Yes, actually, there is one or two paper that has tried hippocampus to try to test, study Alzheimer's disease particularly. I have done hypothalamus, brainstem, cortex. What else? Spinal cord recently. We're vascularizing the spinal cord, same as the MRBO that we're doing. So that's about it.
When you're doing the spinal cord, are you doing the dorsal and the ventral, or how are you doing
Just
that?
this bit so that we can fix the cervical spinal cord region so that we can add it to the brainstem because we have the brainstem. So that area
So
basically.
you're working your way down.
Yeah, working over it.
That's the iPhone 17.
That might be iPhone 20 at this. That will take a while.
What are you using for your extracellular matrix? Are you using like a matrigel or are you?
So for the
Produce.
cortex, we use Matrigel.
Okay,
For this one, it is not, this is some kind of a mixture between, we had to test a lot of things.
yeah,
So this has human fibronectin, collagen 6 and collagen 9s. And this is all human, so one times and very expensive products.
all right,
Have you tried directing the neurons like semaphorin or something like that?
Yes, we're doing that right now with the hypothalamus. We're trying to make sure it goes in the right direction when we attach it to the cerebral cortex. Yeah.
you mentioned N-acetylcysteine.
Yes,
Since running that, have you started taking N-acetylcysteine?
and I do, well.
So I asked in my PhD lab, my PI swore by N-acetylcysteine. He did breast cancer research and found that N-acetylcysteine was extremely, like, it's antioxidant
Yep,
effects
are very good.
are very good. You mentioned neuroprotective. He was looking at it from a different angle and yeah, swore by it.
So at the end of the class, everybody's gonna eat blueberries.
There you go. Very nice.
Yes,
So this is kind of like cloning, right? So to what extent is it legal? Like, when does it become brain then?
oh, I hate this question. I get asked by this question all the time. So yes, since the multi-region brain organoid, as close as we got to a fetal brain, this is a common question that comes up. But now also think your brain is billions of neurons, 86 billion neurons on an average. This is millions. So it's literally, I don't know, very tiny, miniature size. I don't think it, or I would say, in my opinion, it is a miniature version of a brain, but not a complete brain. And your brain also has the lymphatic system, the immune system, the proper circulation, all of that. So, and all of those things help meninges, the skeletal system. So I don't think we're close to an actual brain-like structure or even near there. So right now we're far away from it becoming a human brain, but we're somewhere sort of as a reductionist model getting as close as we can.
And it lacks any sensory input.
Yes, but we can give a sensory input. We can get the electric currents and stimulation. People are trying, so Thomas Harting's lab is interested in organoid intelligence.
It's better to do this than do it on monkeys.
Yes, and that too. Yes.
So you mentioned some of the stuff you wanna do, you can't do because you don't have a pump, you don't have a way to actually create flow in the
Yeah.
organoid. So do you have ideas for that? Are you thinking of like just a tiny little electrical pump, or do you wanna build a little heart and then stick that on your?
It's not as easy as that, but so yes, for now, we're going to install a peristaltic pump. That's the idea, so we're going to do that. And then we need these little tubes to be stable enough to take that pressure.
Yeah.
So we're doing that right now. Then my 3D printing expert is doing that or trying to do that. And we don't want to build artificial. So we want to keep these themselves, but give them the support they need. Now your heart question is very interesting because, I mean, the heart itself has a lot of nerves. So the heart is controlled by the autonomic nervous system. The autonomic has parasympathetic and sympathetic neurons. So that's how you would control the heart. I don't know if anyone has done that yet. Maybe someday. Yes.
I know you touched on this before, but I'm a little bit confused. When can you direct the way that it grows? And if so, does communicating or using signaling with one part of that brain encourage growth more of that side?
So which brain are we talking about?
This
Multi-region.
this multi-region, right? Yeah.
So actually the vasculature, this, I was again talking to Greg about it, it was that if you have, so what we did was, because we keep the media at the end the same, which is just neurobasal and N2B27, which is a very generic, with endothelial factors, very generic media for organ or brain-like structures. We don't try to redirect it after we fuse them. Then we just let it mature or grow, expand it. But one of the things that I found and I published in the paper was, because it's modular in dimensions or size, is that if I have the cerebral and the endothelial only and I take the midbrain out, it doesn't affect cerebral organoids maturation. I mean, it makes it more healthy and all of that, but it doesn't affect, more synaptic plasticity or more cortex-like cells, or layer 5 starts developing. That doesn't happen with the cortex. We saw that, we tested it in various ways, we didn't see anything.
But it is more healthy, like it's functioning more actively.
More healthy, more active, but that's about it. But when I do it the opposite, brainstem and the endothelial, it actually helps the hindbrain development. We saw a lot of HOX B3, D3 patterning, transcription factors and transcription increase in expression once we added the endothelial. So the endothelial component apparently is necessary for hindbrain development.
Adding like new layers and building more onto it.
Yep, it started developing all these, or it didn't say so developing faster, it started developing hindbrain specific markers that are necessary for hindbrain patterning, which is HOX P3 and HOX P3 expression.
Okay. Thank you.
So that, and then this endothelial structure releases a paracrine signaling, and there were 6 factors we found that I published in the paper that helped to do that.
All right. Let's give one more round of applause. I think it's 3:15, so perfect time. Thank you all for coming.